Welcome back to BE544. In Lecture 1, we grounded ourselves in the fundamentals of histopathology, from specimen collection to microscopic diagnosis. In Lecture 2, we transitioned into the digital era, exploring how all slide images are generated, stored, and accessed through scanners, file formats, and public repositories like the CIA. Today, in Lecture 3, we move from theory to practice by introducing QuPath, which is a powerful open-source software platform that bridges digital pathology and computational analysis. QuPath enables pathologists and researchers to view, annotate, quantify, and analyze whole slide images with precision. Critically, it also supports integration with machine learning workflows, making it an ideal environment for prototyping AI-driven diagnostic tools. During this session, you will learn how to navigate QuPath's interface, detect tissue regions, segment cells, extract quantitative features, and prepare data for downstream modeling. This hands-on foundation is essential, not only for understanding how AI system consumes histopathology data, but also for developing interpretable, clinically relevant algorithms. So let's begin our exploration of QuPath. First, we will need to download QuPath and install it on our computer. So you will go to qpath.github.io or Google the word QuPath and open the first link that will navigate you to this website. You have different options to download the QuPath depending on your operating system, whether it's Windows, Mac, or Linux. So in this case, we will download it for Windows and start installing it. This interface will pop up once you open the installation, click on Next, and then select the suitable QuPath installation directory or keep it as default and press Next. You can toggle the shortcut or keep it as default and press Next, and then press on Start Installation. It may take a couple of minutes to be installed. Once it's done, press on Finish. Once you open QuPath after installation, this notice will pop up. You can accept, and also you can always accept by selecting this option and accept. And also this window will pop up to select the theme. So let's keep it system theme and keep the settings as default, and we don't need it to show on each startup of the application. So we will unselect this option and press Get Start. Also, it will ask you for opening the extension manager. So let's keep it remind me next time and press No. This will be the interface of the QuPath application. This black region is the visualization or the viewer of the slide, as we are going to see after a while. Here is a toolbar for the different options, and this panel is for the properties of the project, image annotations, hierarchy, workflow, and we are going to navigate on most of these options during this session. So first we will need to create a project, and inside that project we will import and add whole slide images from any dataset you can download or from the dataset you are working on. So first select the project tab and select create project. So let's create a project and call it lecture 3 project and press select folder. Now we have a project and we need to add images inside this project so that we can work on these images. So from add images, you will need to drag drop images here or choose files and select the different images. As mentioned in the previous lecture, you can download public datasets and work on them. So you can download any dataset from the TCIA and start working on it. So for now, let's assume we have a dataset, for example, BACH dataset from the breast cancer channel, which is a public dataset that can be used. Here in this project, I will drag 5 slides from 1 to 5 to explain the different options of the QuPath. Here you are selecting the image type, and in our case, these images are H&E slides, which is defined in the data source. And this option asks you if you need rotation or not. So keep it by default, no rotation, and press import. Now it will start to process the different whole slide images and create the pyramidal view for each of them. As you see in the left panel, after processing all of them, there are different thumbnails or small images for each of them. To open any of them, you need to double click on the whole slide image, and this will be the view of the whole slide image that you clicked on. Simply, you can left click on any region and drag to a specific region, or you can use the mouse wheel to zoom in and zoom out inside this pyramidal view. So let's assume that I need to focus on this region, so I'm using the mouse wheel and zooming in inside this region. As mentioned, there are different magnification levels, and the highest one is the one that you can zoom in at until there is no more zooms. This will be the highest resolution. As you see here, we are zooming out right now until we reach a reasonable zoom, which is here 20 micrometer. And as you see here, you are seeing different nuclei, cytoplasm, boundaries, and fats. So to work on the specific details, you are targeting the highest zoom or the highest magnification level. And if you need an abstract view or a global view, you can zoom out and get insights on a specific region from an abstract view. Once you select this image, you can move to the second tab, which is called image, to get the different details for this image. This is the name, the location, the magnification level, which is 20x, the width and height in pixels and micrometers, the uncompressed size, which is about 8, and the pyramidal view, we have 1x, 4x, and 16x. And these are the coloring vectors for the stains. And as we mentioned, there is a hematoxylin and eosine, and for each of them there is a specific vector that reflects the color and other properties regarding the hematoxylin or eosine. Also, you can right click and go to display and select brightness and contrast, and here you can change the brightness of the slide, just in case if there is a specific slide where its appearance is not reasonable and they need to be changed. This is the original. So if I selected hematoxylin, the hematoxylin view will be displayed only. If I selected the iosine only would be selected. You can select also other channels, like I need only the blue. So this is the blue channel. So let's return again to the original view. You can select auto so that it can handle, depending on the histogram or the image, the brightness and the contrast. And if you changed the brightness and contrast, as you see here, we are increasing the contrast and the brightness and darkness here. And if you messed with these values, you can press on reset. It will return to the original view. Also, you can select invert background, and in this case, it will invert the background, which is a white color, into a darkish or black color. Also, you can select show grayscale to convert the image from RGB into grayscale, and all of these options can be used so that you can handle the image according to the task you are working on. Now let's move to the annotation and selection. As you see, this is the abstract view as mentioned, and dealing with the whole slide image is not reasonable because there are background, which is here, there are fats, and if you are dealing with a project that targets specific cells or cells with tumor or cells with malignant or benign tumors, then you need to select these regions and extract it later and work on them specifically. So we need to learn how to annotate a specific region or specific regions so that we have the annotations, we have the coordinates of each of them, and using Python or any scripting language, we can extract those images or those ROIs and work on them. So for example, we can extract different regions regarding normal cells, other regions for tumor, and build a machine learning model that can classify between the normal and the tumor. Also, you can make it multi-class classification by selecting some regions for normal, some regions for tumor, let's say benign, and others for malignant tumor, or other grades of the tumor depending on the dataset you are selecting, and build a multi-class classification pipeline and model to classify later between the different categories. So zoom in to a specific region. Let's say that we need to annotate this region. So as mentioned, later we can gather those ROIs using any scripting language. So go to annotations. You will find an empty list because we didn't create any annotations. And we have here different colors or categories. And also you can add your own class by selecting and pressing on the plus sign. Let's say, for example, I will create a region for fats or a class for fats. And also you can select the color by selecting the color from here, or double click and select the color from here. So let's keep the default color as is. Let's create another class with the name nuclei and keep the color as is. As you see in this bar, we have different annotation options such as the rectangle, the circle, oval, line, and so on. So for example, if you selected the rectangle here, and let's say we need to annotate this region like that, you will see the different properties such as it's a rectangle, the XY coordinates, the area, and the perimeter of this region. If you double click outside this region of interest, you will find the color reflecting the class, which is here selected the tumor because it's right here and it's right here in the annotation or right here in the rectangle. You can change the type of the class by right click and set properties and select a class for that. Let's say nuclei, and you can also change the color from here or to a different color as you wish. You can set a name for the annotation, let's say A1, and you can add a description for that annotation and press apply. You will find here this is A1, the annotation, which is here A1 nuclei. To delete it, you can simply select it and press delete and confirm on deleting this object. If you did that for each annotation, it will be tedious. So if we need now to start annotating a set of nuclei, and we need the program to auto-select and auto-set the nuclei category, then simply click on nuclei and press on auto-set. So once you select the rectangle and start the annotation, rectangle again, start annotating this region, you will have now two annotations reflecting the nuclei. And let's say you selected this region by mistake to be in the nuclei, whether you right click and change the properties of it, or simply select this one, which is reflecting the misclassified region, and then select the correct category. Let's say, for example, tumor, and press on set select. Now, this region will be tumor, this region will be the nuclei, and this region will be the nuclei. Again, if you double click outside the region, you are deactivating it. If you double click inside the region, you are activating it, and you will get here different properties regarding this annotation. Also, let's back to nuclei here and select the oval or the ellipse here and select this region, again, this region, again, this region. Now we have 6 annotations, 1 for tumor, 5 for nuclei. You can also create a closed polygon annotation by selecting this option, and this will be more focused. So simply we can zoom in this region to annotate it. So left click and keep the left clicking to select and surround the nuclei. For example, here, now we have a polygon, and these are the properties for it. Also, we have an open loop polygon. So for example, one click here, one click here, one click here, one click here, and return again to the original point. One click here, one click here, and to cancel for the last point, double click from the left click. Now we have another annotation. There are also 2 helpful tools. One is called brush annotation and another called wand annotation, which is here. Let's start with the brush one. And in the brush annotation, it will look like a brush So let's select this fat and press here auto set, ensure that it's activated, and press on the brush. And if you click and move your mouse, it will start to create an annotation like a brush, as you see here. So here we are trying to fill this region like that. And you can also increase the annotation region by trying to find the exact boundary of the white region or the fat, like that. Once you complete the annotation, you can release the left click. Now we have the polygon that reflects the fat, which is here. This is also the one tool as mentioned, and it is more intelligent. So let's say now we need another fat object. So left click and keep the left click, and it will automatically find the surrounding region. So you are just adjusting the location of the mouse, and it will try to spread to find the boundary automatically, as you see here. Let's move to this region and try again. Left click, and it will automatically try to find the region like here. Once you get closer to the boundary, it will start to find the boundary and spread, as you see here, like that. Now we have an exact object. You can refine it by getting closer to the boundary and try to avoid any inner points like that by getting closer towards this region, as you see here. Now we have the polygon using the one tool. The same for nuclei. Auto set, sure it's activated. Zoom into this region. Once you click, it's automatically selected. The same here, the same here, the same here, here, here. Let's move to another portion from the image. Let's say I need this region, this region, this region, and so on. Now, as you see, we have different annotations. You can group these annotations using the hierarchy. So, for example, let's annotate this whole region like that, as you see here. And let's say that we need to group these all nuclei into a specific region. So simply create a rectangle that surrounds those objects, and you will find it also in the hierarchy, which is here. So simply right click on this rectangle and select the annotations and select insert into hierarchy. Once you do that, you will now have this annotation or this object that includes 7 inner annotations, as you see here in this hierarchy. So simply you can group different objects together into regions or categories or bigger categories or parent categories, such as what we did here. The same if we did here by adding a rectangle, let's say for this region, and right click and insert into hierarchy. You will find here this object has 3 inner annotations, as you see here. So now you know how to apply the annotations and the hierarchy. Also, you can change the opacity of the view by adjusting this wheel like that. This is the lowest opacity and this is the highest opacity. And you are doing so just in case you need to make it more transparent so that you can see behind the annotation. Also, you can press on Shift F to fill the annotations, and it will be helpful if you change also the opacity of those like that. This is the highest opacity and this is the lowest opacity, and through it you can see the inner parts or the whole slide image. Also for annotation, as you see, we did select different annotations, but let's assume that we need to annotate this region to be tumor, for example, but I don't need to select the nuclei. So simply, let's select the nuclei, go to annotations, nuclei, and select the nuclei first, something like that. And here, if you did a mistake, you can press on Ctrl Z and revert the selection. So if I selected this by mistake, so that you can press on Ctrl Z, it will be reverted. So let's select it. Now I need to select the outer region, which is here, without selecting this inner nuclei. So simply, I will go to tumor, for example, and start annotating. So by default, it will annotate this region But if you pressed Alt Shift, it will deselect. So if I pressed on Alt Shift during my annotation here and made deselection inside this region, it will deselect it. So I'm here again, I will continue annotation and press deselect, it will deselect. So I need to do annotations here like that. So all nuclei are included. And now I can deselect this region, deselect this region. I will continue my annotation, which is something like that. I will deselect this region. It will automatically deselect it. The same here, the same here. So now I have a polygon that contains this region, excluding the nuclei. And to get sure that we are correct, I press on Shift F to fill the annotation to get ensure that the region excludes the nuclei. Now let's move to a tool called Estimate Stain Vectors, which can be found in the Analyze and select Estimate Stain Vectors. And the QuPath Estimate Stain Vectors tool enables accurate color deconvolution in bright field histopathology images by simply identifying the specific stain vectors for hematoxylin and leucine, which will later improve the reliability of downstream quantitative analysis such as cell detection, classification, and intensity measurements by separating stains objectively, reducing the variability due to differences in staining protocols or scanner settings. So simply we can, here in this application or using scripting language like Python, try to find the vectors using this tool for hematoxylin and leucine and propagate those vectors to all of other images so that we can ensure that all of the images have the same vectors, and this will help to reduce the variability because for sure you can gather data from different places or different scanners, which for sure may get different settings or protocols, and hence each of them will have different vectors. But by unifying those vectors, which will help later improve the reliability in your project. So once you select the estimate stain vectors, this option may pop up. So let's press yes, and you will find here the stain vectors, original and the current, and by default, both of them are similar. So let's select auto. It will recalculate the stain vectors, and these are now the current vectors. By clicking OK, it will apply the current vectors on the image. So this is the original one, and by pressing OK, it will ask for a name for the new stain vector. Let's keep it by default and press OK. The new vectors will be applied on the image. Now you can take those values, which now will be found here in hematoxyl and leucine under the name H and estimated. You can take these two vectors and apply them to other images. For example, let's get the hematoxylin right now. Press OK. Go to another image. Double click on it. If there are anything need to be saved here, it will ask first. So save the first SVS file. Now we are in the second image. Go to image. You can change these values like that to the new values. You can also get the vector for the leucine. Go to image, get the value from here. Okay, go to the second image and change the value to the new one like that. Also, you can automate this process by simply go to the original one, which is here. These are the values. Simply, you can go to the workflow. You will find the different command history here that you did. And this is the last one that we did, which is set color deconvolution stains. And these are the details. So we can create a script from that by pressing on create script and getting only the last script here and removing the remaining of them. For readability, go to edit, wrap lines. So the lines will be wrapped like that. Now we have the two vectors inside this command. You can save it and apply it to any SVS file or use that later with Python to automate the process for all of other slides instead of doing that manually. So first, save the script. Let's say estimate stains. It will be stored as a Groovy file. Save it. You can run it to ensure that's working. Now we can go to the third slide like that, which by default have these two vectors. Now we can go to automate script editor. You will find the scripts in this project. Press on run. As you see, the vector changed. Do the same for the fourth slide. Run the script. The same for other slides. Now you know how to estimate the stain vectors using the estimate vectors tool. And based on these two vectors, you can propagate these two vectors on all of the images by simply creating a Groovy script by going to the workflow and getting the last command, which is applied by the stain tool, store it as a Groovy file, run it in the target slide. It will be applied on that slide.